Hello, I'm Hannah.
And I'm Ashlyn, and our project is investigating Clathrin-mediated endocytosis in Drosophila dendrites.
The internalization of extracellular cargo, such as growth factors and other signals, is required for cell survival. Without internalizing these signals, cells go into programmed cell death. When looking at the dendrites of neurons and how they may be receiving these cargo from other cells, such as skin cells, which are shown here, there are various different possibilities as to where this internalization could be taking place. Cargo internalization could be occurring at the cell neuron contact sites. It could also be occurring at energetically favorable sites in the dendrites, or internalization could be happening in the cell body and axons of the neurons, and the cargo could be then transported to the dendrites. Clathrin-mediated endocytosis is the primary form of cargo internalization in neurons. And I want to highlight the formation of clathrin-coated pits, which is shown here, in which clathrin, which is the key endocytic player, forms a coat around the incoming cargo to form a vesicle. And then during the scission step of endocytosis, dynamin forms a collar around the incoming vesicle to pinch it off and allow for its full internalization. Previous studies in our lab show that clathrin particularly localizes to dendritic branch points in stationary and mobile punctate forms. By temporarily blocking endocytosis using a temperature-sensitive dynamin mutant, although the localization of stationary clathrin puncta does not change, there was a great decrease in the presence of mobile clathrin puncta. And because clathrin localizes to dendritic branch points and temporarily blocking endocytosis causes changes in clathrin activity, this leads us to hypothesize that dendritic branch points are active sites of endocytosis. Particularly, we're asking whether stationary clathrin at branch points are the clathrin-coated pits, and if other endocytic players such as dynamin localize to dendritic branch points. And in order to answer these questions, we use Drosophila model organisms, genetic manipulation techniques, fluorescent tags, and live imaging to study protein localization and dynamics in live Drosophila larvae. We first looked at clathrin activity when endocytosis was temporarily blocked using a dynamin temperature-sensitive mutant, in which we would heat shock Drosophila larvae, and this would induce a dynamin mutation that blocked dynamin activity. We then use fluorescence recovery after photobleaching assay to study clathrin exchange, in which we would bleach a stationary clathrin puncta at branch points using a high-intensity light ray. And if clathrin exchange was occurring, we would see recovery of the clathrin's fluorescence. If no exchange was occurring, we would not see recovery. When endocytosis was active, what we found was that after bleaching a clathrin puncta, its fluorescence recovered quite quickly. However, in the temperature-sensitive mutant where endocytosis was temporarily blocked, after bleaching the clathrin, although we did see eventual recovery of its fluorescence, its recovery time was a lot longer than that of the normal conditions when endocytosis was active. And this can be visualized in this graph here. We then looked at clathrin activity when endocytosis was constitutively blocked. And in order to constitutively block endocytosis, we used a dynamin dominant negative mutant, which completely blocks out dynamin activity and arrests endocytosis at its scission step. When endocytosis was active, we saw again that after bleaching the clathrin puncta, it recovered quite quickly. However, in the dominant negative mutant, when endocytosis was blocked, we saw that after bleaching the clathrin puncta, there was essentially no recovery happening. And these results go to support that stationary clathrin puncta are constantly undergoing exchange. And when we block endocytosis, we are decreasing this exchange. We then looked at how dynamin localizes in neurons. And we found that dynamin not only localizes to dendritic branch points, similar to clathrin, but it also co-localizes with the stationary clathrin at branch points. And both of these results, that stationary clathrin are undergoing exchange and that dynamin is particularly co-localizing with the stationary clathrin, these results support that the stationary clathrin are clathrin-coated pits, and therefore branch points are active sites of endocytosis.
And so thanks to Hannah's research, we now have more confidence to say that these branch points are sites of active endocytosis. And so the big question we have now is why? What makes these sites so special? And so we can see here to the left, a 3D picture of what the typical branch point looks like. And the most notable features are the blue concave curvature regions and the red convex curvature regions. There's also the center regions and the flat edge regions. And looking over to the actual dendrite, the curvatures are highlighted in blue as well. And so as we know from the beginning of our presentation, endocytosis requires a concave curvature to bend in and form that important bag-like vesicle that eventually will be pinched off. And so it makes sense that endocytosis tends to favor these concave regions because they already have this important curvature. And this was something that we saw in previous lab data, that Clathrin tends to localize at the concave curvatures of branch points rather than the convex curvatures of branch points. And this might be because they're more energetically favorable. So the big question, why does endocytosis localize at branch points, leads us to the sub-question, does Clathrin favor branch point concavity, and why? So to answer this question, we decided to use an overexpressed spastin line to reduce the amount of convex membrane curvature, as well as increase the amount of concave curvature. So an image on the left shows a regular dendrite with the red convex regions and the blue concave regions. On the right, we see a spastin line, which tends to make much larger, fatter branch points that kind of merge together. So this limits the amount of red convex curvature, as well as increases the surface area of concave curvature. And we use this to see how Clathrin localization might change. So in this picture, we can see the real-life effects of spastin using confocal microscopy, as well as fluorescent proteins. So on the left is the regular control branch point, and the right is the spastin branch point, which you can see has much fatter tubes and larger branch points than the much thinner ones of the control line. And so looking at the data, in black is the control line, which obviously has a high favorability for the concave curvature of branch points compared to the flat, center, and convex curvatures. And in the gray is the spastin line, which has those wider, fatter branch points. And so we saw a much wider distribution of Clathrin localization, which makes sense because if concave curvature is what's most favorable for Clathrin, if you increase the amount of concave curvature available and decrease the amount of convex curvature available, you're going to see Clathrin appearing in more places because there's more places for Clathrin to be endocytosed, and it's more favorable that way. So just to recap, larger branch points in spastin change Clathrin localization due to the reduced convex curvature. And we plan to further visualize this using mathematical calculations.
Because we have evidence that dendritic branch points are sites of active endocytosis, we can now look to how endocytosis may play roles in other pathways that particularly happen at branch points. Microtubule nucleation is the building of microtubules, which are cytoskeletal proteins important for maintaining the neuron's dendritic arbors. And microtubule nucleation is particularly initiated at dendritic branch points due to the localization of the gamma-tubulin ring complex, which you can see here in the blue, and you can see on the right here. The gamma-tubulin ring complex essentially acts as the template for microtubules to grow. And the initiation of nucleation is controlled by the Wnt-frizzled complex, which is composed of a Wnt ligand bound to a frizzled receptor. And together, the signaling complex recruits various nucleation players to dendritic branch points and eventually gamma-tubulin. And then this allows microtubule nucleation to be initiated. Preliminary data from our lab suggests that the source of Wnt ligand in this complex comes from extracellular skin cells. Once the ligand is bound to the frizzled receptor, however, this leaves us with the question of how is this complex particularly brought to dendritic branch points and internalized? And because we know that dendritic branch points are sites of active endocytosis, this leads us to hypothesize that the Wnt-frizzled complex is actively endocytosed at dendritic branch points, and therefore endocytosis is acting upstream of microtubule nucleation. So in order to test this hypothesis, we studied the localization of a cytosolic nucleation player called disheveled, shown here in pink, and we looked at its localization when blocking various steps of endocytosis. So we first blocked the scission step of endocytosis using the dynamin dominant negative mutant that I discussed earlier. And what we found is when endocytosis is active, disheveled localizes to dendritic branch points in discrete punctate forms, as shown here. And the image on the right shows the saturation of these disheveled puncta, which gives us a good idea of how concentrated the disheveled is at these branch points. When blocking the scission step of endocytosis using the mutant, however, we found that disheveled almost accumulated at these branch points. Although its localization didn't change, the disheveled puncta became a lot more saturated. And this is suggesting that hypothetically, when the Wnt-frizzled complex cannot be fully internalized, when the scission step of endocytosis is arrested, disheveled can still be recruited to dendritic branch points, but it will accumulate there. We next decreased endocytosis altogether using Clathrin RNAi. Clathrin RNAi knocks down the expression of clathrin and in turn decreases clathrin-coated pit formation and endocytosis altogether. When endocytosis was active, we saw that most branch points were occupied by a disheveled puncta. However, when endocytosis was decreased altogether using the Clathrin RNAi, we saw that many branch points were now unoccupied by disheveled, and so disheveled recruitment to branch points generally decreased. And this goes back to support that decreasing endocytosis leads to decreased nucleation machinery recruitment to branch points. And this leads us to the conclusion that endocytosis is acting upstream of nucleation machinery recruitment to branch points. Here's a summary of our key findings in our projects. In terms of future projects, I'll be looking at if cytosolic nucleation machinery, such as disheveled, co-localize with clathrin-coated pits when endocytosis is blocked.
And I plan to really zoom in on concave membrane curvature and determine whether it really does make endocytosis energetically favorable, as well as mathematically modeling this and looking at other classes of dendrites. These are references, and we just like to thank Dr. Rolls as well as other members of our lab for the continued support. Thank you so much for listening.
Shifting gears to the university libraries questions. The first question is, tell us a little more about your research process and what steps did we take to find out about our project. So we both work in Dr. Melissa Rose's lab, where we're really focused on looking at the organization of neurons and how they respond to injury. So for our projects, we are working with a grad student who is looking at microtubule nucleation and events upstream of it. So that's how we sort of got our project topics. Yeah.
Okay, and then for the second question, if you're not at Penn State, would you be able to access the resources provided? Why does this access matter? How do you think people outside of universities access and learn about this kind of research? Personally, I don't think I'd be able to have the same research opportunities if I wasn't at Penn State, especially as an undergrad. I think that Penn State does a really good job encouraging undergrads to get involved in research and provides a lot of opportunities to get into labs. I think that's really unique to Penn State. I think this access is important because we can learn a lot in our fields and really develop those important scientific skills that we'll use later on. I think that people outside of universities probably struggle to get into these sorts of research situations to do research. So I'm really thankful to have this opportunity at Penn State.
And the final question is, how do you see yourself as a researcher in your discipline after completing this project? I know for both of us, working in Dr. Rose's lab and presenting at the undergraduate exhibition really gives us more experience with not only developing our projects based on hypotheses, developing the experimental approaches for them and analyzing our data, but also creating different ways of visualizing and presenting our data in research. So in research posters, for example, like creating different graphs and different schematics to effectively communicate our findings with different audiences. So we kind of do see ourselves as not only better researchers, but better communicators.